Greetings, tutor. Welcome to the Deep Dives. Today, we're not just opening a textbook chapter, are we? We're really trying to peel back the layers on, well, one of the most fundamental aspects of human biology, locomotion and movement. This is such a crucial topic for our class 11th and 12th board exams, and definitely for competitive tests like NEET. I'm really eager to solidify my understanding here, and I know many of you listening probably are too. So, yeah, let's embark on this journey.
Namaste. Absolutely. And you're right. This chapter, Locomotion and Movement, it truly is a cornerstone of human physiology. It's not just about, memorizing facts. It's really about understanding the incredible symphony of systems that allows us to interact with the world. And yes, it definitely carries significant weight in your biology exams and those competitive tests. So we'll break down every concept step by step, making sure there's absolute clarity and, you retain it. Let's start with something foundational, drawing a clear distinction between movement and locomotion. This is often the very first conceptual hurdle students face.
Okay, that sounds like a perfect starting point. Really fundamental. So what's the core difference then between general movement and something more specific like locomotion? They often seem, well, kind of interchangeable, but I guess there's a key nuance.
You're spot on. There's definitely a nuance, and getting it is key. To start, movement is, well, it's a hallmark, a defining characteristic of all living beings. Seriously, think about it. From the simplest unicellular organisms, tiny microscopic things, right up to complex humans like us, everyone moves. It's truly universal. Yeah. So in its broadest sense, movement covers any change in position. Could be a body part, could be the whole organism. For instance, think about a tiny amoeba, the protoplasm inside it, the jelly-like stuff. It's constantly streaming, flowing internally. That's movement. Or maybe a paramecium using its little hair-like cilia just to wiggle in place. That's movement too. A hydra, right, with its tentacles, it can extend and retract them. And even in our own bodies, just blinking your eyelids, chewing with your jaws, moving your limbs when you gesture, even talking with your tongue, these are all examples of movement. It's a very, very broad term. Covers pretty much any kind of shift or change in position, big or small, internal or external.
Right, okay. So movement is clearly this huge category. Makes sense. What then specifically defines locomotion? How does that narrow things down?
Excellent question. And this is where that critical distinction comes in. Locomotion is a specific type of movement. The key differentiating factor here is that locomotion is a voluntary movement, and it results in a change of place or location of the entire organism. So if you're walking, say, from your desk to the kitchen, or running a race, climbing a tree, or think about a bird flying or a fish swimming, these are all forms of locomotory movements. Why? Because they involve changing your overall position in space. Your whole body shifts from one place to another. It's about, covering distance. And what's also quite interesting to note is that the structures animals use for locomotion, they aren't always just for locomotion, they often multitask. For example, in that paramecium, those same cilia that push it through the water also help sweep food particles towards its mouth, the site of pharynx. Similarly, a hydra uses its tentacles not just for moving, but also for capturing prey. That's its main job, actually. And even for us, our limbs, arms, and legs, there are primary tools for locomotion, like walking or running, right? But we also use them constantly for simple movements, like changing posture while standing still or gesturing while we talk. So it shows that while movement and locomotion are distinct concepts, the biology behind them, the structures, they're deeply interconnected, often doing double duty. Very efficient, really.
I see. Okay, that makes it much clearer now. So if I'm getting this right, is it correct to say that all locomotion is movement, but not all movements are locomotion? This sounds exactly like one of those statements they love putting in board exams, to check if you've got the concept straight.
Precisely. You've absolutely nailed that takeaway. And you're right. It is a classic statement, pops up frequently in both board exams and competitive tests like NEET. Let's just cement this point for everyone listening.
Yeah.
Every single time an organism changes its overall place, any act of locomotion, it inherently involves movement of its parts or its whole body. You can't walk without moving your legs, right? Simple as that. However, a movement like, say, blinking your eye or your heart beating rhythmically inside your chest, or even that subtle protoplasm streaming within a single cell, these don't change your overall location in the environment. They are definitely movements, yes, but they are not locomotion. Here's an analogy that sometimes helps make it stick for exams. Think of a car. When the car drives down the road, its entire location is changing, so it's both moving and locomoting. But if that same car is just idling at a traffic light, maybe the engine is vibrating or the windshield wipers are moving. Those are movements, but the car itself isn't changing its spot on the road, so it's not locomoting, see? helps reinforce that crucial difference. Movement is broad, locomotion is specific. It's about changing your physical location.
Oh, the car analogy really does simplify. It makes perfect sense. I can definitely see why that's a key concept. Okay, so moving on to the why. Why do animals, including us, actually do this? What are the main reasons, the driving forces behind locomotion? What's the evolutionary point?
Right. Another common short answer question for your exams, this one. And it really gets to the heart of survival. Animals perform locomotion for several really crucial reasons, all tied directly to, well, staying alive, thriving, and reproducing. Firstly, and maybe most obviously, it's the search for food. Animals have got to move to find nourishment, whether that's grazing, hunting, or just foraging around. Secondly, it's vital for finding shelter, a safe place to rest, hide from predators, or protect themselves from bad weather like extreme heat or cold. Thirdly, locomotion is absolutely critical for finding a mate. You need to move to reproduce to keep the species going. Fourth, related to that, is finding suitable breeding grounds, places where they can safely lay eggs or raise their young without too much danger. Fifth, it lets them move towards favorable climatic conditions. Think about bird migrations flying thousands of kilometers just to escape the winter. Amazing stuff. And finally, a really, really big one, often life or death, is to escape from enemies or predators. Got to be able to run, swim, or fly away from danger. So those 6 points pretty much cover the main drivers for locomotion across the animal kingdom. Shows just how essential it is for life.
That covers the why really comprehensively. Yeah, it makes you appreciate how purposeful even small movements are. Okay, now let's zoom in a bit more specifically into us, the human body. Are there different types of movement happening at a more fundamental level, like at the cellular level?
Excellent point, because before we can even think about running a marathon or waving hello, our individual cells are already doing their own little dances. So yes, our human body shows 3 main types of movement, particularly at the cellular scale. These are amoeboid movement, ciliary movement, and muscular movement. Let's take a look at each one, focusing on how they work and where we find them in our system. First up, amoeboid movement. As the name pretty clearly suggests, this type of movement is remarkably similar to how a single-celled amoeba gets around. In our bodies, you find this happening in specialized cells. Think macrophages and leukocytes. These are types of white blood cells, key players in our immune system. The mechanism is fascinating. It works by forming pseudopodia, which literally means false feet. Imagine temporary finger-like bits pushing out from the cell membrane. These are formed by the active streaming of the protoplasm, the internal cytoplasm within the cell. It basically makes the cell kind of flow forward like a blob. And crucially, the cell's internal scaffolding, the cytoskeleton, specifically tiny protein threads called microfilaments, they're deeply involved in making this happen, driving the flow. Now, why is this important in our bodies? It's absolutely critical for our immune responses. It lets these immune cells actively crawl through tissues, engulf nasty things like bacteria or cellular junk, and move towards sites of infection or injury. They're like our body's little defenders, patrolling around.
That's a great visual for amoeboid movement, like little cellular crawlers.
Right.
And ciliary movement. That's about those small hair-like things on some cell surfaces, right? I remember that from earlier biology.
Absolutely, you got it. Ciliary movement involves exactly that. The coordinated rhythmic beating of cilia. These are short, fine, hair-like projections sticking out from the cell membrane, almost like tiny oars working together. You'll find ciliary movement in most of our internal tubular organs, the ones lined by what's called ciliated epithelium tissue with these cilia. A classic example, really important for your health, is your trachea, your windpipe. The coordinated beating of the cilia there acts like a microscopic escalator, constantly sweeping upwards. This action helps remove dust particles, pollen, any foreign stuff we might accidentally breathe in with the air. It's a vital protective mechanism for our lungs. Another key place for ciliary movement, the female reproductive tract. Here, the cilia lining the fallopian tubes beat in coordination to help move the ova, the eggs, from the ovary towards the uterus after ovulation. Without this ciliary action, the egg's journey would be much harder, making fertilization less likely. And it's worth remembering, maybe from Chapter 8 on cell biology, that cilia and flagella are actually specialized outgrowths of the cell membrane. They share a similar core structure, that 9+2 arrangement of microtubules. And just for completeness, while it's not for general body movement in humans, flagellar movement is another type. It involves longer whip-like structures called flagella. The key example in humans, vital for reproduction, is the swimming motion of spermatozoa. Beyond humans, you see flagella used for moving water in sponges and for locomotion in protists like euglena. So these cellular movements, they're like silent microscopic ballets happening inside us all the time.
Right. And that brings us to muscular movement, which I guess is the main one we notice day to day, the one that powers most of our visible actions.
Exactly. Finally, muscular movement is indeed the most common and, well, obvious type of movement we exhibit. It's what mostly comes to mind when we think about moving. This type fundamentally relies on the unique contractile property of muscles. Our muscles are specialized tissues, sort of biological engines designed specifically to shorten forcefully. They generate tension, pulling on things, and this contractile ability is harnessed for moving our limbs, our jaws for chewing, our tongue for speaking. Really, most of the complex actions performed by humans and most multicellular organisms. Now, here's a key insight, important for understanding complex actions, not just simple twitches. Locomotion, especially in complex organisms like us, isn't just about muscles working alone. It's a real team effort, a symphony, you could say. It needs perfect, highly coordinated activity from 3 interconnected systems. First, the muscular system itself, providing the raw power, the force. Second, the skeletal system, giving a rigid framework, the levers for the muscles to pull against. And third, the neural system, our brain and nerves, sending the precise electrical signals to control, initiate, and coordinate everything. It's this intricate, almost instantaneous coordination that lets us do incredibly complex things. Catching a cricket ball, playing a musical instrument, dancing. Understanding this seamless integration is really critical for your exams. It highlights the holistic nature of how our bodies work.
That holistic view really puts it into perspective. Not just muscles in isolation, but a whole coordinated system. Okay, now that we get the types of movement, I'm really keen to dive deep into muscles themselves. They seem like the prime movers, the real workhorses, as you said. What are their basic characteristics? And how do we sort of classify them?
Good. Now that we appreciate the different ways movement happens, let's indeed focus entirely on muscles. Because they truly are the workhorses, the dynamic engines driving movement in our bodies. Muscles are a specialized tissue, and interestingly, primarily of mesodermal origin. That's an important little detail for NEET. Remember, it refers to their embryonic source, the middle germ layer. They make up a really significant chunk of our body weight, usually around 40 to 50% in an adult human. That tells you how crucial and abundant they are. Now, what makes muscles so special? What allows them to do what they do? It comes down to 4 key properties. These often pop up as definitions in exams. First, excitability. This is their ability to respond to a stimulus, usually an electrical signal from a nerve. Think of it as them being able to get switched on. Second, contractility. This is their defining feature, the ability to shorten forcefully, generate tension, and pull, converting chemical energy into mechanical force. Third, extensibility. This means muscles can be stretched, pulled longer than their resting length without getting damaged, important for reaching or stretching fully. And fourth, elasticity. This is their ability to snap back, to recoil to their original resting length after being stretched or contracted, like a rubber band. These 4 properties working together allow muscles to perform their incredibly diverse and powerful functions. Now, for classification, muscles are grouped based on different things: where they are located in the body, what they look like under a microscope, and how their activity is controlled voluntarily or involuntarily. Based on location, which is a very common way to classify them, we identify 3 main types: skeletal muscles, visceral muscles, and cardiac muscles. Let's break down each type now. Distinguishing between them is a frequent exam topic, especially for NEET. First, skeletal muscles. As the name clearly tells you, these muscles are closely attached to the skeletal components, your bones. They're the muscles you consciously use to move your arms, legs, torso, maintain posture, any physical action you decide on. Under a microscope, they show a distinct striped appearance, like little stripes. That's why they're also called striated muscles. Furthermore, and this is key, their activities are generally under voluntary control from the nervous system. You consciously decide to move them, like lifting your arm or walking. Because of this conscious control, they're also known as voluntary muscles, their main jobs. Locomotory actions and changing body postures. Think about the effort in doing a squat or jumping, that's skeletal muscle. Next up, visceral muscles. These are found lining the inner walls of our hollow visceral organs. Things like the alimentary canal, your digestive tract, the reproductive tract, the urinary bladder, other internal organs not directly linked to the skeleton. Unlike skeletal muscles, visceral muscles do not show any striations under the microscope. They look smooth, uniform. Hence, they're commonly called smooth muscles or non-striated muscles, a critical difference. Their activities are entirely not under voluntary control. You don't consciously manage digestion or the movement of gametes, so they're known as involuntary muscles, their vital functions. Assisting food transport through the digestive tract, that wave-like motion called peristalsis, and helping gametes move through the genital tract, they work quietly behind the scenes. And finally, cardiac muscles. As the name says, Those are the muscles that make up the wall of the heart. Appearance-wise, cardiac muscles are striated, like skeletal muscles, but they have a unique feature. Their cells assemble in a branching pattern. This branching helps form an interconnected network, essential for spreading electrical signals quickly and coordinately through the heart. Regulation-wise, like visceral muscles, cardiac muscles are completely involuntary. The nervous system doesn't directly control their basic beat. You can't consciously stop your heart. They have their own intrinsic rhythmicity. They generate their own electrical impulses to contract a property called autorhythmicity. This lets the heart beat continuously, tirelessly throughout life without conscious thought, a truly vital function.
Okay, got it. Skeletal. Voluntary, striated. Visceral. Involuntary, smooth. Cardiac, involuntary, striated, and branched with that intrinsic rhythm. That's a super clear distinction, definitely helpful for exams. Now, let's zoom right in on the skeletal muscle specifically. What's its microscopic structure like? This seems complex, but probably crucial for understanding how muscles actually contract.
Excellent summary. Perfect grasp of the types. Now, yes, let's examine skeletal muscle in detail. This part is absolutely critical for understanding the mechanism of muscle contraction, which is, a major topic and often a bit tricky. I'll break it down into its building blocks so you can visualize it clearly. Okay, so an organized skeletal muscle, like your biceps, isn't just one solid chunk. It's cleverly layered. It's made of many muscle bundles, also called fascicles. Think of them like bundles of wires wrapped together. These fascicles are then held together by an outer layer of tough connective tissue called fascia. Now, look inside one fascicle, you'll find many individual muscle fibers. These fibers are the actual muscle cells, long cylindrical things. Each muscle fiber, being a cell, has its own special plasma membrane called the sarcolemma. It's like the muscle cell's skin. Inside the sarcolemma is the cytoplasm, which we call sarcoplasm. And a very unique thing here, often an MCQ point. A muscle fiber is a syncytium. This means the sarcoplasm contains many nuclei, not just one like most cells. It happens because muscle fuse during development. Another critical bit inside the sarcoplasm is the specialized endoplasmic reticulum called the sarcoplasmic reticulum. Now, this is super important for contraction. The sarcoplasmic reticulum is the main storehouse of calcium ions, K++, remember that. Calcium is the key trigger for contraction. Think of it as the cell's calcium vault. A key visual feature of the muscle fiber is the presence of loads of parallel filaments inside the sarcoplasm. These are the actual contractile bits called myofilaments or myofibrils. And when you look at a myofibril under a microscope, you see that striated appearance, the pattern of alternating dark and light bands. This pattern comes from the precise arrangement of two key proteins, actin and myosin. Let's define these bands. The light bands contain only actin. Actin filaments are thinner, so they're called thin filaments. These light bands are the I band or isotropic band. The dark bands, on the other hand, contain mostly myosin. Myosin filaments are thicker, so they're thick filaments. These dark bands are the A band or anisotropic band. Now, within these bands, there are more important landmarks. Right in the center of each light I band, there's an elastic line called the Z line. It cuts the I band in half. The thin actin filaments are firmly attached to these Z lines, anchoring them. Looking at the dark A band, the thick myosin filaments in it are held together in the middle by another line, a thin fibrous membrane called the M line. So the A and I bands are arranged alternately along the myofibril, creating that striped look. Now, for one of the most crucial definitions for your exams, especially NEET, the sarcomere. The part of the myofibril that lies between two successive Z lines is the functional unit of contraction. This segment is called a sarcomere. When a muscle contracts, it's basically thousands and thousands of these individual sarcomeres shortening simultaneously all along the myofibril. That's what makes the whole muscle shorten. It's like tiny engines lined up. If you picture a sarcomere when the muscle is relaxed, the edges of the thin actin filaments partly overlap the ends of the thick myosin filaments. But there's a central part of the thick myosin filament that isn't overlapped by actin. This region, a lighter zone within the A band, is called the H zone. H for Henson's disk, or heller, meaning brighter. What happens to this H zone during contraction is an important clue. Need an analogy to pull this complex structure together. Imagine a long railway track that's your myofibril. Each wooden sleeper across the track is like a Z line, marking off a segment. The segment of track between two sleepers. That's your sarcomere, the basic functional unit. And the metal rails within each segment are like the filaments, actin, and myosin. They're the parts that actually slide past each other when the muscle contracts. How they're arranged and interact within each tiny sarcomere dictates how the whole muscle works, whether you're just twitching a finger or lifting something heavy.
Wow, okay, that is a lot of components. But the sarcomere as the basic unit, like tiny engines lined up, that makes perfect sense. It really helps visualize it. So could you elaborate a bit more on the proteins themselves, actin and myosin? What are their individual structures like, since they're so central to the whole contraction thing?
Excellent question. Yes, understanding the exact structure of these contractile proteins, actin and myosin, is absolutely fundamental to getting how muscle contraction works at a molecular level. Let's break each one down. Their details are often tested. Okay, first, the actin-thin filament. It's not just a single strand. It's actually made of two F filamentous actins twisted around each other in a helix, like two strings of pearls wound together. Now, each F actin strand itself is a polymer, meaning it's built from many individual smaller monomer units called globular actins. So picture lots of spherical G-actin proteins linked together to form one F-actin filament, and then two of these F-actin filaments twisting around each other. But that's not all for the actin filament. There are two other very important proteins closely associated with the F-actin along its entire length. These are tropomyosin and troponin. Tropomyosin consists of two filaments that run right alongside the F-actins, kind of nestled in the groove between the two F-actin strands. And troponin is a complex protein, not a continuous strand, but distributed at regular intervals along the tropomyosin. Now, the crucial role of troponin is this super important concept. In the resting state, when the muscle isn't contracting, a part of troponin physically masks or covers the active binding sites for myosin on the actin filaments. This masking action is absolutely key. It prevents myosin from binding whenever it wants, which would cause constant, unwanted contraction. Think of troponin as the gatekeeper, keeping things locked until the signal calcium arrives. Now, let's look at the myosin-thick filament. This is also a polymerized protein built from many repeating monomer units. These individual monomeric proteins are called meromyosins. So many meromyosins join together to form one thick myosin filament. Each meromyosin unit itself has two key parts: a distinct globular head with a short arm sticking out and a long straight tail. The head and the short arm together are called heavy meromyosin, or HMM. The tail part is called light meromyosin, or LMM. Functionally, that HMM component, especially the globular head and its short arm, is incredibly important. This part projects outwards from the main shaft of the myosin filament at regular intervals and angles. This projecting part is what we call the cross arm or cross bridge head. This is what actually reaches out to grab onto actin. The globular head itself is amazing. It's highly active functionally. It acts as an active ATPase enzyme, meaning it can break down ATP, adenosine triphosphate, and release energy. This energy release is absolutely vital for powering the muscle contraction. Plus, the same globular head has specific binding sites, one for ATP itself, and critically specific active sites for actin, allowing it to link up with the thin filaments. So the myosin head, think of it as the muscle's tiny engine and its grappling hook all rolled into one.
Okay, wow. The masking by troponin, keeping things blocked, and the ATPase activity of the myosin head, providing the energy and the hook. Those seem incredibly important parts of the puzzle. Right now, for the big one, the core mechanism itself. How does muscle contraction actually happen? It's the sliding filament theory, right? That's the one we really need to nail for the exams.
Precisely. You've hit the nail on the head. The sliding filament theory is indeed the most widely accepted model, the best explanation we have for muscle contraction. And it basically states that muscle fiber contraction happens not because the filaments themselves get shorter, but because the thin filaments, actin, slide over the thick filaments, myosin. Imagine you're pulling on a rope tied to a wall. The rope itself doesn't shrink, but as you pull, the distance between your hands and the wall decreases. It's a similar idea here. Okay, let's break this down step by step for crystal clarity. This sequence is super important and frequently tested. Step 1, initiation of signal. Okay, first things first. Muscle contraction always starts with a command, a signal from our central nervous system, the CNS, your brain, and spinal cord. The signal travels down a nerve cell called a motor neuron until it reaches the muscle. And it's important to know, a motor unit is defined as one motor neuron plus all the muscle fibers it connects to or innervates. The crucial connection point, the synapse, where the motor neuron talks to the muscle fibrous membrane, the sarcolemma, is a special junction called the neuromuscular junction or motor end plate. This is the communication hub. Step 2, neurotransmitter release and action potential, right? So when that nerve signal, the impulse, arrives at the end of the motor neuron at the neuromuscular junction, it triggers the release of a chemical messenger. This neurotransmitter is called acetylcholine. Acetylcholine quickly crosses the tiny gap, synaptic cleft, and binds to specific receptors on the muscle fibrous sarcolemma. This binding triggers an action, basically an electrical signal or impulse that starts on the sarcolemma and then spreads rapidly across the entire surface of the muscle fiber like ripples on a pond. Step 3, calcium release. Okay, now this action potential doesn't just stay on the surface, it travels deep into the muscle fiber's interior through tiny tunnels called T-tubules. When the action potential reaches that specialized storage vault, the sarcoplasmic reticulum, it causes a massive and very rapid release of calcium ions, Kbm++, from the reticulum out into the sarcoplasm, the muscle cell's internal fluid. This sudden flood of calcium into the sarcoplasm, remember, this is the critical trigger. Without this calcium surge, nothing else can happen. Contraction can't start. Step 4, unmasking of active sites. Now, calcium ions are floating around everywhere in the sarcoplasm. These Cali++ ions then bind to a specific part of that troponin protein complex, which sits on the actin filaments. This binding of calcium to troponin causes a crucial conformational change, basically. The troponin molecule changes its shape. This shape change, in turn, pulls the tropomyosin strand aside, shifting its position. And by moving tropomyosin, it effectively removes the masking of the active binding sites for myosin on the actin filaments. The sites are now exposed, actin is ready to interact, the gate is unlocked. Step 5, cross-bridge formation. Okay, now the stage is set. The myosin head, remember it acts as an ATPase, comes into play. It breaks down an ATP molecule, ATPA, ADP plus pi plus energy. This released energy energizes or cocks the myosin head, putting it into a high-energy sort of ready position. Now, this energized myosin head can bind firmly to the newly exposed active site on the actin filament. When the myosin head attaches to actin, this connection is called a cross-bridge. Think of it as the myosin head reaching out and grabbing onto the actin filament securely. Step 6, power stroke, sliding of filaments. Right after the cross-bridge forms, the real action happens. The myosin head, using the energy it stored from breaking down ATP, undergoes another shape change. It pivots or swivels inward. This pivoting action pulls the attached actin filament toward the center of the A band, towards the M line. This pulling movement is called the power stroke. It's the actual force-generating step. Now remember, the actin filaments are anchored to the Z lines. So as the actin filaments are pulled inwards by the myosin heads, the Z lines themselves are also pulled inwards, closer together. This inward movement of the Z lines results in the shortening of the sarcomere. And as we said, the shortening of countless sarcomeres is what causes the entire muscle fiber, and thus the whole muscle, to contract. It's important to picture what happens to the bands during the shortening. This often comes up conceptually. The light I bands get reduced in width. They become narrower because the actin filaments are sliding further into the A band region. However, the dark A bands retain their length. The myosin filaments themselves don't shorten, they just act as the track along which actin slides. And the H zone, that central region of the A band where there was no actin overlap, it also shrinks, or can even disappear completely during strong contraction as the actin filaments slide further inwards. These band changes are visible evidence of the sliding mechanism. Step 7, cross-bridge detachment and recocking. Okay, after the power stroke, the myosin head releases the ADP and inorganic phosphate pi that were left over from the ATP breakdown. It goes back to a lower energy state, but initially it stays attached to actin. Now, for the myosin head to detach, a new ATP molecule must come along and bind to it. This binding of fresh ATP is absolutely critical. It's what causes the cross bridge to break. The myosin head lets go of the actin filament. Once detached, this newly bound ATP is immediately hydrolyzed, broken down, again by the myosin head's ATPase activity. ATP, ADP plus pi plus energy. This energy release air cocks the myosin head, returning it to its high energy, ready to bind state, preparing it to grab onto actin again, maybe further down the filament for the next cycle. Step 8. Cycle repetition and relaxation. So this whole cycle, cross-bridge formation, the power stroke pulling action, detachment when new ATP binds, and recocking as ATP is broken down repeats over and over again. It continues as long as two crucial things are available: enough Cas plus ions in the sarcoplasm to keep the actin binding sites unmasked, and a continuous supply of ATP to fuel the myosin heads and allow detachment. Now, how does the muscle relax? Well, the signal from the nervous system stops. No more acetylcholine release. Without the nerve signal, the muscle cell actively pumps the Cas plus ions back into the sarcoplasmic reticulum using special calcium pumps. This pumping process itself requires ATP energy. As the calcio plus concentration in the sarcoplasm drops significantly, calcium ions detach from troponin. Once calcium leaves troponin changes shape back, allowing tropomyosin to slide back and remask the active sites on the actin filaments. The binding sites are covered again. With the binding sites blocked, myosin heads can no longer form cross bridges or existing ones break. The actin filaments then passively slide back to their original resting position, likely helped by elastic forces within the muscle structure. This causes the Z lines to return to their original positions, the sarcomere lengthens, and the muscle fiber relaxes. Phew. It's quite an intricate, beautifully regulated cycle.
That's incredibly detailed, Tutor. Wow. Breaking it down like that, step by step, and the tug-of-war analogy, it really makes the whole process of muscle contraction much, much clearer. Thank you. Now, I've also heard about, muscle fatigue, like after really intense exercise, and also about different types of muscle fibers suited for different activities. Can you touch upon those next?
Absolutely. Good points. Those are also important concepts, frequently showing up in exams and very relevant if you think about sports science or even just understanding why you feel tired after a tough workout. Yes, after intense or prolonged physical activity, muscles can definitely experience fatigue. Let's talk about muscle fatigue. So this condition happens when muscles are repeatedly activated without enough rest, or critically, without an adequate supply of oxygen. The main cause of muscle fatigue, especially during really strenuous activity where you can't get enough oxygen in quickly anaerobic exercise, is the accumulation of lactic acid. What happens is when oxygen supply can't keep up with the muscle's energy demand for normal aerobic respiration, the muscle cells switch gears. They start breaking down glycogen, stored glucose, anaerobically to make ATP. A major byproduct of this anaerobic process is lactic acid. As lactic acid builds up, it lowers the pH inside the muscle cells, making them more acidic. This acidic environment messes with various things like enzyme function and how calcium binds, ultimately making it harder for the muscle to contract efficiently. That leads to that familiar burning feeling and the inability to maintain force fatigue. Okay, now about the types of muscle fibers. Our muscles aren't all the same. They vary in their makeup and how they work, optimized for different kinds of tasks. They're often classified based on their content of a special red-colored pigment called myoglobin. Myoglobin is similar to hemoglobin in your blood, but it's found inside muscle cells. It stores oxygen right there in the muscle, giving muscles their reddish color and providing an immediate oxygen reserve. So, based on myoglobin content, we generally talk about two main types. First, red fibers, also known as aerobic muscles. These muscles have a high myoglobin content, making them look distinctly reddish. Because myoglobin stores oxygen, these fibers have oxygen readily available right inside the cell. They also contain plenty of mitochondria, remember those are the cell's powerhouses, and they have a rich blood supply delivering more oxygen. These mitochondria use the abundant oxygen very efficiently for ATP production through aerobic respiration. This means red fibers are great at producing energy sustainably, allowing them to contract for long periods without getting tired quickly. So they're perfectly suited for continuous, prolonged activity that requires endurance. Think about the postural muscles in your back holding you upright all day, or the leg muscles of a marathon runner. Second, we have white fibers, also called anaerobic muscles. These muscles have very little myoglobin, so they appear paler, more whitish. They also have fewer mitochondria compared to red fibers, but they tend to have a higher amount of sarcoplasmic reticulum. This allows for very rapid release and reuptake of calcium ions, which means their contractions can be very fast and powerful. White fibers mainly rely on anaerobic processes for energy because they don't have much stored oxygen and fewer mitochondria for sustained aerobic work. This makes them ideal for quick, powerful, but short-duration bursts of activity, like the muscles a sprinter uses for an explosive start or the muscles used for lifting a very heavy weight just once or twice. So you see, the structure and the way they produce energy is perfectly matched to their job. It's a really neat example of biological optimization.
That distinction between red and white fibers is really clear.
Yeah.
And definitely relevant for understanding things like marathon running versus sprinting. Makes total sense. Okay, so we've got the coordination from the neural system, the force from the muscles, but none of it would work without that solid internal framework, right? Let's move on to the human skeletal system. Now, our body's amazing scaffold.
You're absolutely right. The coordination comes from nerves, the power from muscles, but without a strong yet somewhat flexible internal framework, we'd just be, well, a blob. That framework is the skeletal system. It's fundamentally composed of a network of bones and also some specialized cartilages. It does much more than just hold us up. It's quite a marvel. Its main jobs: providing structural support for the whole body, giving us our shape, offering crucial protection to vital organs, think skull protecting the brain, rib cage shielding the heart and lungs, very important. And critically for movement, it aids in movement by acting as levers for muscles. Muscles pull on bones, and bones pivot at joints. That's how we move. Try chewing without jaw bones or walking without leg bones, impossible. Beyond movement, bones are also where blood cells are produced in the bone marrow. and they act as a major storage site for calcium and phosphate, minerals vital for many body functions. Now, it's important for exams to distinguish between bone and cartilage within this system. Bone is a specialized connective tissue. Its matrix is very hard and rigid, mainly due to deposits of calcium salts, especially calcium phosphate. That's what gives bone its strength. Cartilage, also a specialized connective tissue, has a matrix that's slightly pliable, more flexible. This is due to chondroitin salts and more water content. While not as hard as bone, cartilage provides cushioning, flexibility, and support in places like joints, your nose, your ears. A very common NEET MCQ, a real classic. The exact number of bones in an adult human. The answer is 206 bones and a few cartilages. Remember that number, 206? The entire skeleton is usually divided into two main parts for study, the axial skeleton and the appendicular skeleton. This division helps us understand the central core versus the limbs. Let's start with the axial skeleton. As the name suggests, this part forms the main longitudinal axis of the body. Think of it as the central pillar. It comprises 80 bones in total. The axial skeleton includes the skull, the vertebral column, backbone, the sternum, breastbone, and the ribs. Let's dive into the skull. It's complex, very strong, made of two sets of bones totaling 20 bones. These are the cranial bones, 8 of them, and the facial bones, 14 of them. The 8 cranial bones fit together like pieces of a helmet, forming the hard, protective cranium that surrounds and protects your brain. They're joined by immovable fibrous joints called sutures. The 14 facial bones form the front of your skull, your eye sockets, nasal cavity, jaws, giving you your facial structure. Additionally, there's a unique single U-shaped bone called the hyoid bone. It sits at the base of your mouth-throat area. What's special? It's the only bone that doesn't directly connect or articulate with any other bone. It's held by muscles and ligaments crucial for swallowing and speech. Also part of the skull count, deep inside each middle ear are 3 tiny bones, the ear ossicles. These are the malleus hammers, incisius anvil, and stapes, stirrup. So 3 in each ear, making 6 total. And the stapes, that's the smallest bone in the entire human body. A great trivia fact for exams, one specific point. The skull connects to the first vertebra, atlas, using 2 bony bumps called occipital condyles. Because there are 2, the human skull is described as dicondylics. Okay, moving down from the skull, we have the vertebral column, your backbone or spine. It's made of 26 serially arranged bones called vertebrae running down your back, dorsally placed. It extends from the base of the skull down to your pelvis, forming the main support for your trunk. Each vertebra has a hole in the center, the neural canal. All these canals line up to form a channel that protects your delicate spinal cord. The first vertebra, right under the skull, is the atlas. It articulates with those occipital condyles, allowing you to nod your head yes. The second vertebra is the axis, which allows rotation, like shaking your head no. The vertebral column isn't just one rigid rod, it's segmented into 5 regions, which gives it flexibility. Starting from the top. 7 cervical vertebrae in your neck, C1, C7. Then 12 thoracic vertebrae in your upper back, T1, T12. These connect to the ribs. Below that, 5 lumbar vertebrae in your lower back, L1, L5. These are the largest and bear most of the body's weight. Then comes the sacral region, which in adults is 1 fused bone, the sacrum, formed from 5 vertebrae fusing together. And finally, at the very bottom, the coccygeal region, also 1 fused bone, the or tailbone, typically formed from 4 small vertebrae fusing. So what does the vertebral column do? Protects the spinal cord, supports the head, and provides attachment points for ribs and back muscles. Crucial for posture and movement. Next in the axial skeleton, the sternum. This is the flat bone right in the middle of your chest, ventrally located, your breastbone. It anchors the ribs at the front. And then the ribs, we have 12 pairs of ribs, so 24 total. Each is a thin, flat, curved bone. At the back, dorsally, they connect to the thoracic vertebrae. At the front, ventrally, most connect to the sternum. An important feature, each rib is bicephalic, meaning it has 2 connection points at its dorsal end, making the attachment to the vertebrae very stable. Ribs are classified into 3 types based on how they attach at the front. True ribs are the first 7 pairs, 1 to 7. They connect dorsally to vertebrae and ventrally directly to the sternum via strips of hyaline cartilage. Vertebracondylar ribs, also called false ribs, are the next 3 pairs. 8, 9, 10. They connect to vertebrae at the back, but at the front, they don't reach the sternum directly. Instead, their cartilages join up with the cartilage of the 7th rib, so an indirect connection. Floating ribs are the last 2 pairs, 11 and 12. They connect to vertebrae at the back, but they have no ventral connection at all. Their front ends are free, hence floating. They help protect the kidneys. Together, the thoracic vertebrae, the ribs, and the sternum form the protective rib cage. It shields your heart and lungs and also helps with breathing mechanics.
That's a lot of bones just in the axial skeleton. Really helpful to get that clear count and location for everything. Okay, so what about the rest? The bones in our arms and legs. and the bits that connect them to the axial skeleton. That's the appendicular part, you said.
Yes, absolutely. You're right on track. The appendicular skeleton is everything else, basically, the bones of your limbs, arms and legs, along with their supporting girdles. These girdles are like the connection points, the bridges, linking your limbs to that central axial skeleton. And interestingly, each limb, whether it's an arm or a leg, is made of exactly 30 bones. So 4 limbs times 30 bones. That's 120 bones just in your arms and legs. Let's break down the bones of the hand for them first, starting from the shoulder down. You have the humerus, the single bone in your upper arm. Then in your forearm, there are 2 bones running side by side. The radius on the thumb side and the ulna on the pinky side. Moving down to your wrist, you have 8 small blocky bones called carmels, arranged in 2 rows. They allow for lots of wrist flexibility. Next, forming the palm of your hand are 5 longer bones called metacarpals, 1 leading to each finger. And finally, making up your fingers' digits are the phalanges. There are 14 phalanges in each hand, 3 in each of the 4 fingers, proximal, middle, distal, but only 2 in your thumb, proximal, distal. This difference helps give the thumb its special movement. Now, for the bones of the legs, equilum, starting at the hip, you have the femur, your thigh bone. This is notably the longest and strongest bone in the human body, built for weight bearing. In your lower leg, again, 2 bones. The larger main weight-bearing bone is the tibia, your shin bone, and running alongside it is the thinner fibula, calf bone, which mainly helps stabilize the ankle. At your ankle, similar to the wrist, you have 7 bones called tarsals. They form the ankle joint and the back part of your foot. Forming the main arch and body of your foot are 5 metatarsals leading towards your toes. And for your toes, just like fingers, you have phalanges. Again, 14 in total per foot, 3 in each of the smaller toes and 2 in your big toe. 1 extra bone in the leg is the patella, your kneecap. It's a cup-shaped bone, technically a sesamoid bone, meaning it develops within a tendon, sitting over the front of the knee joint. It protects the joint and helps your thigh muscles extend the leg more efficiently. Okay, next, the girdles. These are the crucial structures that literally girdle the body, connecting the limbs, appendicular skeleton, to the trunk, axial skeleton. Each girdle has 2 halves, 1 for each side. First, the pectoral girdle, or shoulder girdle. This connects your upper limbs, arms, to the axial skeleton and allows for that amazing arm mobility. Each half of the pectoral girdle consists of 2 bones, a clavicle and a scapula. The clavicle is your collarbone. It's that long S-shaped bone you can feel at the front of your shoulder. It acts like a strut, holding your arm out away from your body. The scapula is your shoulder blade. It's a large, flat, triangular bone on your upper back, usually lying over ribs 2 to 7. On the back of the scapula is a prominent ridge called the spine, which extends sideways into a process called the acromion. The clavicle connects to this acromion. Just below the acromion, there's a shallow socket called the glenoid cavity. This is super important because the rounded head of the humerus, upper arm bone, fits into this cavity, forming the very mobile shoulder joint. Second, we have the pelvic girdle, or epigirdle. This connects your lower limbs to the axial skeleton. It's much more robust and stable than the shoulder girdle because it has to bear the body's weight. It consists of 2 large coxal bones, also just called hip bones. Each coxal bone is actually formed by the fusion of 3 separate bones during development. The ilium, the big wing-like upper part you feel on your hips, the ischium, the lower back part you sit on, and the pubis, the lower front part. Where these 3 bones meet and fuse, there's a deep cup-shaped socket called the acetabulum. This is where the rounded head of the femur thigh bone fits in, forming the strong and stable hip joint. The 2 coxal bones meet at the front, ventrally, forming a joint called the pubic symphysis. This joint contains fibrous cartilage and allows for a tiny bit of movement, which becomes important, for example, during childbirth.
Okay, so we have the bones for structure, the muscles for force. But how do we get that smooth, coordinated movement where bones meet? That must be where joints come in, right? These connection points seem incredibly important for actual motion.
Precisely. You've identified the final crucial piece of the puzzle for movement itself. The unsung heroes of flexibility, you could say. With bones as levers and muscles providing force, how do we get actual, controlled, flexible articulation? That's exactly the role of joints. Joints are basically defined as the points where bones connect to other bones, or sometimes where bones connect to cartilages. They're absolutely essential for all types of movements involving our skeleton. Walking, running, bending, twisting, all depend on joints functioning correctly. Joints act as fulcrums, or pivot points, for the forces generated by muscles. A muscle contracts, pulls on a bone, and that bone moves around a joint like a hinge. Now, the amount of movement possible at a joint varies a lot. It depends entirely on the joint structure and what its job is in the body. For classification, joints are grouped into 3 main structural types based on what holds them together and, importantly, how much movement they allow. These are fibrous joints, cartilaginous joints, and synovial joints. Let's look at each one. Knowing their characteristics and examples is vital for exams, especially those matching questions in NEET. First, fibrous joints. The key feature here, they do not allow any movement. They are completely immovable fixed joints. How? The bones are essentially fused end to end, held tightly together by dense fibrous connective tissue like tough threads. The classic example is the sutures between the flat bones of your skull, like the parietal, temporal, frontal bones. These sutures lock the cranial bones together rigidly to form that protective cranium for your brain. No movement needed, just solid protection. Second, cartilaginous joints. These joints allow for limited movements, just a small amount of give or flexibility. Here, the bones are joined together directly by cartilage, either hyaline cartilage or fiber cartilage. A great example is the joints between adjacent vertebrae in your spine. Those intervertebral discs made of fiber cartilage connect the vertebral bodies. The slight movement between each vertebra adds up along the whole spine, allowing you to bend and twist your torso, but without being unstable. Another example is the pubic symphysis we mentioned in the pelvis. Finally, and these are the most relevant for the kinds of movement we do every day, synovial joints. These are characterized by having a fluid-filled synovial cavity between the articulating surfaces of the bones. This cavity is lined by a synovial membrane which produces synovial fluid. This fluid acts like oil in a machine, lubricating the joint, reducing friction, and absorbing shock. This whole setup allows for considerable movement, making synovial joints essential for locomotion and most other complex movements, throwing a ball, typing, everything. They're the most common type of joint, especially in our limbs. Now, it's really important for your exams to know the different types of synovial joints and specific examples. These pop up all the time in matching questions. First, the ball and socket joint. Here, a rounded, ball-like end of one bone fits into a cup-like socket of another. This allows movement in multiple planes forwards, backwards, sideways, rotation, offers the greatest range of motion. Think shoulder joint, humerus and scapula's glenoid cavity, and hip joint, femur and pelvis's acetabulum. Next, the hinge joint. Just like a door hinge, it allows movement primarily in only one plane, usually flexion and extension, bending and straightening. Classic examples are your knee joint and elbow joint. Then the pivot joint. This allows rotational movement around a central axis. The key example is the joint between the atlas C1 and axis C2 vertebrae in your neck. This lets you rotate your head side to side like when you shake your head no, leaving. Gliding joint or plane joint. Here, the articulating surfaces are mostly flat or slightly curved, allowing for limited gliding or sliding movements back and forth or side to side. Examples include the joints between the carpal bones in your wrist and between the tarsal bones in your ankle. And lastly, the saddle joint. This is a special type where the articulating surfaces are saddle-shaped, concave in one direction, convex in the other, fitting together like a rider on a saddle. It allows movement in 2 planes, biaxial, giving more flexibility than a hinge joint. The prime example is the joint between the carpal bone trapezium and the first metacarpal of the thumb. This gives your thumb its unique opposable movement, crucial for gripping.
That's a super comprehensive breakdown of joints. Their functions, the different types, and those specific examples are really useful. Okay, finally, it's also really important for exams to know about common health problems, right? So could you briefly cover some common disorders of the muscular and skeletal systems, things we should probably be aware of for the exams?
Absolutely. Good thinking. It's important to be aware of common disorders affecting these systems. They often appear in the diseases section of exams, maybe asking about symptoms or causes. Plus, understanding what goes wrong helps appreciate normal function even more. Okay, first, myasthenia gravis. This is a serious autoimmune disorder. That means the body's immune system mistakenly attacks its own tissues. Specifically, it targets the neuromuscular junction, that nerve-muscle communication point. It destroys acetylcholine receptors on the muscle side, so the nerve signal can't properly trigger the muscle. This leads to progressive fatigue, weakening, and potentially paralysis of skeletal muscle. It often starts with eye muscles, then spreads. Second, muscular dystrophy. This isn't just one disease, but a group of inherited conditions. They're characterized by the progressive degeneration of skeletal muscle. The muscle tissue wastes away over time, losing function, often replaced by fat or scar tissue. These are mostly due to genetic disorders, faulty genes leading to problems with essential muscle proteins, like dystrophin. Third, tetany. This involves rapid spasms or uncontrolled wild contractions in muscles, often causing cramps. The main cause is very low levels of Cal++ calcium ions in the body fluid. This really highlights how critical calcium is for normal muscle control. When calcium is too low, nerves become overly excitable, firing signals constantly, leading to those spasms. Fourth, arthritis. This is a very general term, simply meaning inflammation of joints. There are many types, like osteoarthritis from wear and tear or rheumatoid arthritis, autoimmune, but they all involve joint pain, stiffness, swelling, and reduced movement. It can really impact mobility. Fifth, osteoporosis. This is typically an age-related disorder, more common as people get older. It's characterized by decreased bone mass and density. The bones become weak, brittle, and porous, increasing the chance of fractures, even from minor falls. A common factor, especially in women after menopause, is decreased levels of estrogen, as estrogen helps maintain bone density. And finally, gout. This is another type of inflammation of joints, but with a specific cause. It's due to the accumulation of uric acid crystals within the joint space. If uric acid levels in the blood get too high, it can crystallize into sharp, needle-like structures that deposit in joints, causing sudden, severe attacks of pain, redness, and swelling. The big toe is a common sight, often related to diet. Okay, so that brings us to the end of our deep dive into locomotion and movement. We've covered quite a bit. We started by nailing the difference between movement and locomotion, explored the different cellular movements, amoeboid, ciliary. Then we spent a good chunk of time on muscle structure, right down to the sarcomere, and importantly, the step-by-step mechanism of contraction, that sliding filament theory driven by calcium and ATP. After that, we mapped out the skeleton, both axial and appendicular, listing the key bones and their numbers. And finally, we classified joints and looked at some common disorders. Remember, all these parts are interconnected. It's a system. Muscles need bones and nerves. Movement needs joints. For your exams, really focus on the definitions. Know the structures, muscle proteins, actin, myosin, troponin, triple myosin, and the sarcomere inside out, and that step-by-step process of muscle contraction. Know it cold. Understand the roles of calcium and ATP. Also, bone names and numbers, especially the total 206, are classic NEET questions. And be ready to match joint types with examples and their allowed movements. So, my advice now. Revise this material thoroughly. Maybe even go over it again tomorrow. Try drawing that sarcomere diagram yourself from memory. Label everything. See if you can explain the band changes during contraction. And definitely practice writing out the steps of muscle contraction without looking at your notes. Explain each step clearly. And crucially, practice past year questions on this chapter. That's the absolute best way to see how these concepts are tested and lock them in. If any doubts pop up while you're revising or if something still isn't clicking, please feel free to reach out. Keep up the excellent work. Your hard work and dedication will definitely pay off.